Assalamualaikum and greetings. My name is Danish Ofahmi bin Muhammad Jeffrey O. Matrix number A174944. As you can see, there is a large image of a fly. This is because today I'm going to present you my research proposal, which is about the spatial distribution of sarcosaprophagous scuttleflies under the Diptera order and Phoridae family in UKM Bangi as environmental and forensic indicators using QGIS. My research is mainly supervised by Dr. Raja Muhammad Zuha and co-supervised by Encik Hukil Sino. First off, let me introduce what are sarcosaprophagous insects. The term may sound scary and lengthy, but it actually means insects feeding on dead or decaying flesh. The scuttleflies focused in this research are considered as sarcosaprophagous because once a human or animal is dead, scuttleflies are one of the earliest species to arrive on the carcass, especially in concealed environments. When a biotic component dies, it will undergo decomposition. From a forensic perspective, the larvae or adult fly found on the carcass provides information about how much time has passed after death, commonly known as postmortem interval or PMI. Next, as you can see, spatial distribution is the arrangement of individuals in a species in a habitat or between habitats. For example, when we talk about the spatial distribution of monkeys in UKM, we are observing the presence of monkeys throughout the whole UKM. Spatial distribution provides us information such as richness and abundance, with richness meaning number of species in an area, meanwhile abundance refers to the number of individuals per species. Now that I've briefly explained the main characters in this research, I'd like to address the problem statement where there is a lack of research about the association between scuttleflies and their habitat. We know that scuttleflies are rich in species and abundant in various habitats, but I'd like to draw a relationship between scuttleflies species and their habitats. The spatial distribution of the species will be visualized using QGIS, and the data will be used as forensic and environmental indicators. So, how will this information be used in forensic and environmental contexts? The spatial distribution of scuttleflies in various habitats can assist in PMI estimation. It can show whether a body has been moved from the location of death to another location because the distribution of scuttleflies may vary in different types of habitats, such as an urban setting or forested setting. Meanwhile, the richness and abundance in the scuttleflies can be used as an indicator of biodiversity in a specific habitat. This is the research questions. So, what is the diversity status of sarcosaprophagous scuttleflies at selected habitats in UKM Bangi? This question focuses on the general distribution of scuttleflies in the mentioned habitats. Secondly, are distributions of sarcosaprophagous scuttleflies attributed to different types of habitats in UKM Bangi? This question leads to the relationship between the distribution of scuttleflies to the types of habitat. The main goal of this research is to determine the spatial distribution of sarcosaprophagous scuttleflies in UKM Bangi as forensic and environmental indicators using QGIS. So these are the specific objectives. There are three. The first objective is to list down all scuttlefly species collected from all sampling sites. The second objective is to draw out the association between the scuttlefly species and the types of habitats. The third objective is to create a visual representation of the spatial distribution of scuttleflies throughout the sampling sites in UKM Bangi using QGIS, which is a geographic information system software. The hypothesis being tested in this study is that the distribution of sarcosaprophagous scuttleflies is associated with geographical habitats in UKM Bangi. We are looking forward to a certain pattern of species present in different types of habitats at a higher affinity. This is the research design. This research is considered as an experimental study using the type of habitat as factor. This will determine the sampling sites in UKM and consequently affect the observed distribution of scuttlefly species. The locations chosen are UKM Forest Reserve, Tasir FCAP, and College Dato' On. The reason for choosing these locations is that all three locations are different settings, with UKM Forest Reserve being a forested setting, Tasir FCAP is an outdoor water body setting, and College Dato' On is an urban setting. The sampling instrument that will be used is a white-colored pan trap. This is the most efficient and inexpensive method to trap the samples. The study duration will be 3 months, where the sampling process will take place in 1 month, repeated thrice in all 3 sampling sites. The data collected will be the number of species and insects trapped in accordance with the geographical information of the sampling site. This map shows all 3 sampling sites in UKM Bangi. All 3 locations are accessible and have different distances to nearby forests. UKM Forest Reserve is a forest itself, Tasir FCAP being not too far away from a forest, while College Dato' On being the furthest away from a forest. And this is my sampling method. This is the white pan trap. So first off, approximately 10 to 20 grams of fresh chicken liver will be used as bait and placed in the middle of the pan to attract nearby insects, specifically scuttleflies. As the water and detergent creates a mixture with decreased surface tension, any insect landing on it will sink and drown. These trapped insects are considered as the sample and will be collected every 3 days. The pan trap will be secured under a metal cage to prevent sample loss due to scavengers such as dogs, lizards, or any other animals. A research was once conducted by Campbell and Hanula stating that white color is preferred by Phoridae or scuttlefly family over other colors, and that the white pan trap captured the highest number of samples. In order to achieve the first and second objective, white pan traps will be set up to collect samples in the chosen sampling sites. Then, the samples collected will be dissected for species identification by observing its taxonomic characteristics. Once the samples are identified, they will be tabulated and distributed according to its habitat. Here, any relationship between the distribution of scuttleflies and the type of habitat will be evident. Finally, the distribution will be visualized using QGIS. And these are the expected results in this study. This is an example of the distribution of samples according to its respective habitat. As you can see, there's rainforest fragment, sugarcane plantation, sandy beach, and urban area. This is the example. And this is the final product of the distribution using QGIS. As you can see, it's a map. It looks like a map. And this is my Gantt chart. Currently, I have covered the proposal writing, research design, and literature review. I'm on my way to begin manuscript writing, and I look forward to conduct a pilot study, data collection, and analysis starting this August. And this process will go well, and the manuscript will be done by next February. Here are the list of references that assisted me in designing this research and provided useful information for this study. That is all for my research proposal presentation. Feel free to ask any questions and feedback. I am excited and looking forward to conduct this research. Thank you and stay safe.